The ground is shifting beneath our feet and beneath the roots of the crops that feed us. Our global food system is a sprawling, intricate network sustaining 8 billion people, now facing a challenge of unprecedented scale. The climate is warming faster than our agricultural systems can handle. This is not a distant threat. It is a clear and present reality. Scientists have issued stark, data-driven warnings. The IPCC says, for every degree Celsius of warming, global yields for major crops, maize and wheat, could fall dramatically 7.4% and 6.0%. The delicate balance that lets a seed sprout, grow, and produce grain is being upended by heat, drought, and unpredictable weather. We are already seeing the consequences. For thousands of years, farmers and plant breeders have been humanity's primary innovators in agriculture. They have painstakingly improved our crops through a process of selection and cross-pollination. Selection, cross-pollination. This is conventional breeding. It involves identifying a plant with a desirable trait, withstands a bit less water, survives a mild frost, and crossing it with a high-yielding crop. The breeder then plants the resulting seeds and spends generations selecting the offspring that successfully combine high yield with the new trait. This method has been the engine of agricultural progress, giving us the bountiful crops that have fed civilizations. But its fundamental limitation is time. It is a slow, methodical, and patient process. The development of a single new crop variety through conventional breeding is an epic undertaking. It can easily take a decade, and often longer, from the initial cross to the moment a farmer can plant the new seed in their field. Show timeline diagram, long multi-year timeline with branching paths and dead ends. The process is fraught with uncertainty and biological constraints. In the face of this challenge, a new set of tools has emerged. These tools fall under the umbrella of gene editing. The most famous and versatile among them is known as CRISPR-Cas9. To put it in simple terms, gene editing works like a pair of molecular scissors guided by a GPS. Scientists can design a guide molecule that directs the scissors and enzyme like Cas9 to a very specific location in a plant's DNA. Once there, the enzyme makes a precise cut. The cell's natural repair mechanisms then mend the break, and in the process, a small change can be made. A gene can be turned off, or a tiny alteration can be made to its sequence. This process is fundamentally different from the one used to create what most people think of as older genetically modified organisms, or GMOs. The creation of those organisms typically involves a transgenic process. This means a gene from a completely different species, say a bacterium, is inserted into a plant's genome to give it a new function, like producing its own insecticide. While effective, this introduction of foreign DNA has fueled public debate and regulatory scrutiny. Gene editing, by contrast, most often makes small, targeted tweaks to genes already present in the plant. The two most revolutionary advantages of gene editing are its precision and its speed. Think of conventional breeding as trying to edit a single word in a book by rewriting the entire chapter and then proofreading it over and over again. You might fix the word, but you risk introducing new typos and grammatical errors along the way. Gene editing, in contrast, is like using the find and replace function on a computer. It goes directly to the specific word you want to change and alters only that word, leaving the rest of the book untouched. This level of precision eliminates the problem of linkage drag, the unwanted genetic baggage that slows down traditional breeding. This precision directly translates into incredible speed. As mentioned, developing a new crop variety through conventional breeding can take a decade or more. With gene editing, that timeline can be dramatically compressed. Scientists can introduce a desired trait into an already successful, elite crop variety in a single step. The process of discovery, editing, and initial testing can be completed in as little as 3 to 5 years. In a world where climate patterns are shifting rapidly, this speed allows us to be proactive rather than reactive. we can develop the seeds needed for the climates of 2035, not the climates of 2015. To prepare our crops for a harsher climate, scientists are using gene editing to focus on a handful of critical survival traits. Diagram. Plant with icons pointing to roots, leaves, whole plant. The goal is to build a more resilient plant from the ground up, one that can withstand the key stresses of a warming world. First and most critical trait, water use efficiency. With droughts becoming more frequent and severe, we need plants that can produce more food with less water. Stomata, the tiny pores on leaves that release water. By editing genes that control the stomata, scientists can make plants breathe less and conserve moisture. For example, research on rice has shown that editing a gene called OSDST can help the plant maintain its yield while using significantly less water, a vital adaptation for arid regions. Second major target, heat tolerance. Extreme heat can be devastating for crops, especially during sensitive periods like flowering and grain filling. High temperatures can cause photosynthesis, the process plants use to convert sunlight into energy, to shut down. Rubisco activase. Researchers are using CRISPR to test edits that create more heat-stable versions of these proteins. The aim is to create plants that can continue to photosynthesize and grow productively even during a heat wave, protecting yields when they are most at risk. Third, we must address salt tolerance. To understand the real-world impact of this technology, we can look at specific regional challenges where gene editing offers a tangible solution. Consider the vast, densely populated river deltas of Asia, such as the Mekong in Vietnam, the Ganges-Brahmaputra in Bangladesh, the Ganges-Brahmaputra in India. Rising sea levels and reduced freshwater flow are causing saltwater to creep inland, poisoning rice paddies that millions depend on. A small increase in soil salinity can mean the difference between a harvest and total crop failure. Gene editing is not an abstract exercise, it's a lifeline. Scientists at institutes like Arid are developing rice varieties that thrive in saline conditions. Early field trials show promising results. Some gene-edited lines maintain yield where conventional rice fails. For a farmer earning only a few dollars a day, boosting yields by half a ton per hectare can mean food security and a path out of poverty. In the business of agriculture, time is money. The long development cycle of conventional breeding is a strategic disadvantage in a changing climate. It is also a significant financial burden. The costs are spread out over a decade or more of field trials' labor land use. Gene editing fundamentally alters this economic equation. It requires significant upfront investment in labs and expertise, but it dramatically compresses R&D timelines. That concentration of effort drives more efficient resource use and a faster return on investment. Speed matters against climate change. It enables forward breeding. Scientists use climate models to project conditions 10 or 15 years ahead, then use gene editing to develop future-ready varieties today. This proactive approach helps us adapt before crises hit. Technology, no matter how powerful, is only effective if society accepts it. The history of transgenic GMOs looms large over the introduction of gene editing. The Frankenfood label, born from public anxiety. For gene editing to succeed where its predecessor stumbled, a different approach is needed. One built on transparency, clear communication of benefits, and a respect for public concerns. Recent surveys conducted in 2024 and 2025 show a growing acceptance of gene-edited foods, particularly when the benefits are tangible and clearly explained. When asked about products that offer improved nutrition, reduced food waste, or enhanced sustainability, a significant majority of consumers express a willingness to buy them. This suggests that the public is more open when it solves shared problems: health, cost, or environmental impact. Gene editing is not a magic wand that will solve the climate crisis on its own. It must be integrated with sustainable farming practices, robust soil management, equitable policies, However, it is an exceptionally powerful tool, offering speed and precision unimaginable a generation ago. It provides a viable pathway to help stabilize our food supply on a volatile planet. To harness this potential, we must move forward with intention and strategy. The choice is not between technology and nature, it is between acting and delaying. First, governments must create a clear, predictable, science-based regulatory environment and move toward regulatory harmonization, embracing product-based frameworks and mutual recognition to facilitate trade. Second, investors must fund biological infrastructure, stack multiple beneficial traits, and build local capacity in the global south. Third, researchers must build trust through transparency, open science, and engagement on real farms.